Oké, ik denk dat de meesten weer terug zijn. Most people have returned, so we're going to continue. For the purpose of the stream, please be extra quiet. Okay, I'm gonna start real slow, make sure that in the other rooms people also are aware that we are starting again. Okay, so this is the second lecture. In the first lecture, I'll provide a really general overview of what we're going to do and the two different courses and the logistics of the course. This lecture, I kind of want to prepare you for what to expect on the exam. So this is almost an exhaustive list of what you have to know for the exam. A really important and large part of the course is focused around linear regression, and that's what most of this lecture is going to be about. But of course not everything is linear, so we will introduce a few nonlinear models. And then there are just techniques that are really useful that either aren't a model or they're just something that you add to an analysis you already have, like principal component analysis. That's a method for it's basically regression without an outcome, just exploring is there anything going on in my data. Then there's cross-validation, which as the name suggests is a way to validate that your model is stable and that it is actually giving a good prediction out of sample. Then there's bootstrapping, which is like magic because we can now estimate standard errors and confidence intervals when we can't. And then finally there's regularization, which is again something that we can apply to linear regression models. That's a method to deal with when you have so many explanatory variables that normal methods don't work. Then you would use regularization. There are also other use cases, but that is in a nutshell what we're going to talk about. Yeah, so those are the topics. That's it. The skills that you need to pass the course are basic programming R. Also for advanced, your programming skills don't have to be really well developed. You just need to have a basic idea of the syntax in R. You need to be able to copy a script and then adjust it such that it works for your use case. So a lot of the comments on general research skills, the third year course, students would say, well, I only learned to copy paste. Well, welcome to programming. That is what everybody does, also people who get paid for it. Okay, another really important thing is that you are able to choose a model based on expert knowledge. So what is expert knowledge? It's the knowledge that you have as a biologist, plus the little bit of extra knowledge on how would you frame that in a regression model. So that's one. You have to choose, okay, this model makes more sense from a biological point of view. So I'll give a more concrete example later in the lecture, but let's say that you want to know which treatment is more effective, this one or that one, and you try different concentrations. There's a really good chance that concentration dependence is different for those treatments. You know, that one requires much larger concentration than the other, so you would have to include an interaction. Now, the people who haven't done GRS may or may not have heard of interaction in regression models, but this is what I mean by, if you think about it as a biologist, you need to be able to come up with a reasonable model. We're not going to perform a bunch of tests and then have the computer decide the best model, because that has a lot of problems, as we'll cover in great detail later in the course. Okay, then once you've done that, it may still be difficult to get one final model. You know, you may have a handful that all kind of make sense. So we can have the data answer for us, you know, among a small set of candidate models, which one is the better one. And so that is choosing a model based on statistical measures. So think of AIC, BIC, so Akaiki's information criteria, stuff like that. But also the press statistic, which is a bit more advanced. It's very easy to run with packages, which give you a predictive measure. So you have to be able to interpret those measures. We don't cover many of them. In total it's like 5. And you just have to know which is better, higher or lower. Then you have to be able to interpret model diagnostic. This is very important. When you use a model for inferential purposes, so that means you are interested in the effect size, you're interested in the p-value or confidence interval, you want to answer a question with your model about a relationship, then it is really important that the model that you use is valid for inference. And the first step is, again, to choose a model that makes sense, using your expert knowledge. The second step is to kind of confirm or at least troubleshoot using model diagnostic to make sure that the model that we're using is actually reasonable given the data we have. So that's model diagnostics. Now then the last one is obvious. There's no point in doing any statistics unless you actually know what to do with the outputs. So you have to recognize outputs. Again, not of a very large number of techniques. You have to know what the output of a t-test and a chi-square test looks like, and of regression models. So linear regression, ANOVA, ANCOVA, multiple linear regression, GLM, mixed models. The last one, that's something you haven't had yet, but that's something we're going to cover in this course. Yeah? So that brings me to the main subject of this lecture, namely regression analysis. Because right now you're like, okay, but I'm interested in another technique, or I want to do machine learning, but I'm going to try and convince you that almost everything we do is regression analysis. So regression, why is it called regression? To regress means to go back to a simpler state or a more primitive state. And that is what we're doing in regression analysis, because we're trying to fall back onto some simpler representation of the relationship between the outcome and the explanatory variables. So that's what we regress. So what we're doing, we're trying to get a simple explanation of the data generating process. And that's a very important word because we have a sample. Usually when you do research, you collect observations, or you do an experiment and you record stuff, but you're not interested in the exact findings of your experiment. You're not interested in your sample. You're interested in whatever your sample came from. So that is the biological process, like for example, cancer development. Now, and then any form of regression essentially says that this biological process is a combination of a systematic part, and that's the one that we want to extract, the one we're usually interested in, and a stochastic part, so a random part. Now, the systematic part could be that we have a group that smokes, we have a group that doesn't smoke, so there's a systematic difference between those two. Yeah, or some people may have a genetic disposition for cancer, and others may not. And then there's the stochastic part. That's the difficult part. It's basically everything that we do not or cannot measure, or everything that we cannot measure with infinite precision. So you can think of really minute things like background radiation or random mutations occurring. Even though one person might receive more radiation or carcinogenic substances than another, it is still random where the mutations are going to occur. And this is an influence on the result. So someone may deviate from the model without that really having any meaning. Other environmental effects. So it's really important to understand that regression is about extracting that systematic part, and that the stochastic part is what we make all the assumptions about. Yeah, so statistics is about, very generally speaking, about understanding and expressing the strength of evidence. We'll come back on that in the study design lecture, which used to be day 4, but I think Harald would like me to do it earlier. Then regression is the main tool that we want you to learn during the bachelor course and also during the master course, which is trying to extract a systematic part from a random process like cancer development. What are the things that we can influence? What are the things that we can measure that we know affect the outcome? Okay, so we are simplifying, so that means making errors. And then because of that, there is a very common quote. You'll see it a lot online, and it's very important to keep in mind. All models are wrong. So a model is a simplification, and statistics is not about finding the right model. Your data cannot tell you what the right model is. Your data can only tell you under which model it was more likely generated. So statistics cannot give you the answer to how a process actually works. But that's not necessary, because it can still be very useful. If you can make good predictions, if you can estimate an effect very robustly, then it doesn't matter the model's wrong. It's still useful. If we have a simple model, I'll show it in the next couple slides, for human height in the Netherlands, and the only variable that we put in the model is your sex. Are you male or are you female? Well, there will be a difference. But being male or female is not the only thing that causes your height. So of course the model is wrong, but it is still very useful to estimate the difference. So he goes on to say, George Box, a very famous statistician, that the question you should ask is not whether a model is true, because it never is, but whether the model is good enough for your particular application. Yeah, so this is something, it takes some time to really let it sink in, and there's always, we read all the graded assignments and some of the homework, and there are a lot of people who still confuse after a course what the purpose of statistics is, and they try to say, okay, we rejected this hypothesis, so this is true, or we found that this model is better, so this is how it works. But that is simply too strong a statement. Yeah, that's just what I'm trying to say. Okay, so what then is linear regression? If you were mathematicians, I could summarize the general linear model, as it's called, like this. That's it. It's just any model of this form. And if it fits this, then it works. And all the assumptions we taught you and all the rules and all the tricks, if you were a mathematician, you could derive that just from this. Of course, you have to specify the distribution of the error, but that's it. Yeah, so this is called a matrix notation. It's a matrix of explanatory variables times a vector of explanatory effects, which is beta. But we're not going to cover linear algebra in this course. So this is how you would write that out. This is the same, where we say that every outcome is a combination of the intercept, which is beta 0, plus a sum of explanatory effects times their coefficient. Yeah, so the explanatory effect is x, and the coefficient is beta. And then at the end we have the error, because again, the model's wrong, there's some deviation. Yeah, or in words, the outcome is the intercept plus a bunch of slopes times a bunch of explanatory effects plus some remainder. That is how all linear regression works. If you can somehow reformulate your problem into this form, then you can study it using linear regression. Okay, so here's what that looks like. Here we have a simple linear regression. So that means simple refers to there being one explanatory effect. And we have the model in blue, we have the data in black, and then the intercept is where the explanatory variable is 0. And the slope is the increase in the outcome, so y in this case, when we increase x, the explanatory variable, by 1. So for every unit increase in x, how much does y increase? So that's the steepness of the curve. No curves. With those two things, we have covered the entire model. There's nothing else you can say about this blue line. But linear regression goes much further than just estimating lines. We can do multiple linear regression, where we have an effect in more than one direction. So here's an example. Your systolic blood pressure is caused by many things, but there may be a linear relationship with the age and the systolic blood pressure, and simultaneously there can be a linear relationship with your weight and the systolic blood pressure. And of course, if you go all the way to 0, then it no longer makes sense. But within the range of your data, this could be a good model for estimating someone's systolic blood pressure based on these two explanatory variables. Yeah, and even though this is no longer a line, this is a plane, because it's in two directions, it is still a linear model, because all it is an intercept, which is where it hits the y-axis, and then a slope times the age, and a slope times the weight. So again, this is still linear. Okay, so what I'm trying to say is, as long as you can express a relationship with an outcome in terms of intercept and slope, then it's linear. Yeah? So the math behind it is, we will mention it at times because we want to show you how stuff actually works, but we're not going to ask you to regurgitate any formulas. That's not what this course is like. I just want to show you this is the linear model. If it looks like this, it's linear. So with that in mind, let's go over 4 examples and see which ones are linear, right? Okay, anyone have an idea? Yeah, top left is probably linear. It's an intercept plus a slope times x, right? This is the simple linear model. Okay, let's go to the next one. Yeah, because we can write it as intercept plus slope times explanatory. So that's the model. But this one, is this linear or not? And why? Is it readable? It says 32 plus 5 times log x. Yeah?
It is linear. Yeah.
And why?
Because the slope happens to be 5 log, so you don't get that clean increase of like it just increases differently, basically. But it's
Yeah,
still linear.
exactly. So it's a bit tempting. So she said because it says 32 plus 5 times log x, you know, it's very easy to see a curve and think, okay, maybe it's not linear. But you know, if we rewrite this, you know, I could just say I make a new variable, and that's the log of x. Then all this says is 32 plus 5 times some explanatory variable. So it's still just intercept plus slope times effect. So this is still linear. Yeah, even though it's not a line, it is linear. Okay, how about this one? Linear or not? Let's see if anyone has an idea in the chat. No? Not a problem. Okay, anyone want to give it a go? It's a yes or no question, so you have a 50% chance if you gamble.
I think yes, because you still have just a constant and a variable.
Yeah, so he said, sorry, let me repeat it because of the microphone. He said it's still just a constant plus a slope times a variable. Just because there's more variables doesn't mean it's no longer linear. It says 35, that's the intercept, plus 5, that's the slope, times x, minus 10, that's another slope, times x squared. You know, the x squared, again, is distracting, but if I just give it another name, all this says is intercept plus slope times effect minus slope times effect. Yeah, the minus just means that the size of the slope is minus 10. But it still says intercept plus slope times effect plus slope times effect. Yeah? So that is what linear means. Okay, last one then. Is this one linear? Yes. Okay, here are a couple yes. That's correct. Yeah? Just because I have written a sinusoidal function there doesn't mean it's nonlinear. When would it be nonlinear? Okay, what if I put a number in here like x minus 2? Yeah, that 2, you cannot estimate that linear because it's inside the sinus function. But if you can rewrite it as intercept and slopes times explanatory effects, it's linear. Yeah, so this may seem trivial, but this is a really important mental exercise. First of all, for me to convince you everything is linear regression, but also for you to help you frame your problem and to understand that using this regression framework, you can model a lot of different things, you know. The names that I gave here are a bit misleading because I wouldn't necessarily model it like this, but you could do it. A constant increase, of course you can do it with linear model. But diminishing returns, just fine. Just use a logarithm or a square root or something. A peak, doesn't matter. Just add a polynomial term. Seasonality, okay, we're getting to the part where linear models are not the strongest, but it's still possible just by adding a sinusoidal term. Yeah? So linear models are really, really versatile. And what we're going to teach you both in the basic and in the advanced statistics course is how you can make them even more versatile. So yeah, let me skip a couple slides because we already had these. But first, let me give you one more example in case you're still not convinced everything is linear. This model is clearly linear, right? We have an intercept plus a slope times an explanatory effect. And this time I wrote the error, but whether you write it or not, this is the model. Yeah? But, you know, I can make this variable such that it is equal to 0 if you're anything but male and 1 if you're male. So this doesn't look like a line, you know, it's two lines at two different levels. It can only take on two different values plus error, but still it's linear because the only thing that matters is intercept and slope times explanatory effect. So that means that, we have to specify the error. So let's say that there's a variance of 5 for human length. It's not a random number. I actually pulled this from our World in Data, which is a nice website, by the way. So what does this model say? It says the mean for women is 1.7 plus 0.1 times 0, because this vector, this x, is equal to 0 if you're female, right? So it basically says your height is 1.7 plus error. So what does a plus error mean? It means that, okay, if we convert that to standard deviation, it's a bit easier to work with. Then we would expect female height to be within about 2 standard deviations of their mean. So that means practically that we expect around 95% of women to be, in the Netherlands then, to be within 14 centimeters of 1.7 meters. Yeah, of course I quickly said within in the Netherlands because this model is only useful if we restrict it to where this is more or less true. If you take the whole world, it's probably not a good approximation. Okay, then what does it say for the men? Very simple. It says the mean is 1.7, and this time we add the slope because if you're male, x is equal to 1. So we just add 0.1. That's what it says. Variance, nothing changed. So it's still this variance of 0.05, meaning that we expect men, according to this model, to be within 14 centimeters of 1.8 meters. Yeah, we don't expect all men to be there, but around 95% should be within those bounds. Yeah, so still a linear model. Basically what we did here is, of course, a t-test. Yeah, it's the mean of two different groups. But I'm just showing you the t-test is just, we can frame it in terms of linear regression. And that brings me to the point of this lecture. Almost everything is linear regression. Yeah, these are some of the techniques that I have used the most over the years using biology and biomedical science and clinical science, stuff like that, people I've helped. It's almost always something along these lines what people need. And very rarely are they actually interested in prediction or in exploratory data analysis. Of course, it's useful because it's nice to know your data. And also, maybe you do have a predictive problem. So we do introduce it. But most of what you're going to do is going to be inferential statistics using linear models. Now, in blue here, I have shown the the topics that we cover during the general research skills course. So t-test, I'm going to make a not so bold assumption. I think everybody here knows what a t-test is. ANOVA is just the extension of the t-test to more than more than two groups. Yeah, so in terms of linear regression, we just saw the t-test, the men and the women. We can do it with one dummy variable, just one variable that takes on a 0 if you're one class and a 1 if you're the other class. More groups in ANOVA, doesn't matter, just add more dummy variables and it's still linear. Yeah, simple linear regression, of course linear, multiple linears, of course linear. And COVA is a linear model where you have both these dummy variables, so you have factorial or categorical explanatory effects, and you have continuous explanatory effects. And then GLM, that is where we ended with the general research skills course, and that's also the last regression subject within the basic statistics for master student course. And there we say, okay, all this time we assume that this error is normally distributed. But actually for a lot of data that makes no sense. So let's do a little bit more than just the normal distribution, but let's also consider other distributions as long as they're within a certain family called the exponential dispersion family. Yeah, so these are the most important things you'll learn during basic statistics for master students. And if you can, every day you'll get a new topic, just try to see how it relates to this framework of regression analysis, because that is where most techniques fall into. Now then in advanced statistics, we're going to do something a little bit more interesting, which is called a mixed model. And as Harold already explained in his lecture briefly, essentially that is a model that allows you to relax the assumption of independent data. We can now not only use anything we already learned in linear regression, but we can also use data that are dependent. And that is really useful because let's say you have a clinical trial and you follow your patients for some time and you measure them several times. Of course that's not independent. It's the same people you keep measuring. So it's dependent data, but it is a valid study design. And you would need a mixed model or something equivalent to deal with that, to still get valid inference. Now then the GLMM, it is very tricky to work with. The diagnostics are quite difficult. You don't have to become explicit, but I just want to tell you that conceptually the GLMM is very simple. It's just a GLMM plus a mixed model. Yeah, so now we can do all at once. Now then there's one more topic during the course that is not really linear regression. You can apply it to anything, but we will show you how to use it in linear regression, and that is regularized linear models. Now, so we can do all of the things listed above, but then with regularization. Now, what is regularization? It is a technique that allows you to estimate more explanatory variables than you would normally have enough observations for. So you can do more with less. Okay, then there's other important techniques that I do use a lot, but that are not part of the course. Yeah, so you might come across this during your internship, and of course you can ask those questions, but everything in gray is not in this course. Yeah, so a generalized additive model, that's the first one listed. That is when you know a sensible probability distribution for the outcome, but it is not in the GLM framework. It's not one of those. Yeah, mixed GAM is just combining the two. Then beta regression. Let's say you have an outcome that's a percentage, right? You can say it's a number between 0 and 1. It's either 100%, which is 1, or it's 0%, which is 0. You can do that with beta regression. And lastly, survival analysis. Very, very common in clinical science, but it's a bit too specialized to introduce in this course, and there are a lot of quirks and things you should read about before you delve into survival analysis. Which is a type of GLM specifically for dealing with time until event data. Now, why is it so common in clinical science, and why is it not just called time until event analysis, but survival analysis? Because if I start with 100 patients and I start looking at the number of patients that survive, well, guess what? Some of them don't survive. So you're going to end up with less and less patients, and people may drop out of the study. People may enter the study at a later time, so it's a completely different beast, not part of the course, but it is very interesting and used a lot in clinical science. Yeah? Okay, let me quickly have a look at the chat, just in case. Yeah, no, everybody's just quietly listening, no problem. Okay, let me just briefly recap everyone who did GRS, what the GLM is, and then, also for everyone intending to do basic statistics, this is the big topic we're going to talk about in the third week, the generalized linear model. So what do we generalize it to? We generalize the normal distribution to a family of different classes of distributions which we can choose from. Yeah, so I just gave you the first example, the adult height in the Netherlands, and if we subtract the effect of sex, so let's say we only look at males, then a normal distribution is a reasonable approximation. And actually it's true that most males fall within a certain distance from the mean. But let's say we count the number of birds in a park. You can count 0 birds, or 1 bird, or 2 birds, or any positive integer value, but you can't count 0.1 birds, or -1 bird. So this distribution is not continuous, because it only takes on specific values, it's discrete, and it's non-negative. You cannot have a negative count, right? So that means that when we get closer to 0, the mean, that it's almost always going to be right skewed, yeah, because everything gets pressed into the last bit here, up till the 0, and because of that, it's almost never symmetric unless the counts are really large. Okay, then there's the binomial distribution. The example here is the number of correct answers out of 20 questions. Now, for the moment, let's just ignore the idea that you can give someone half a point, because then it would not be binomially distributed. Let's say you either have it right or wrong every question. Then, at the very most, you can have 20 questions right, because there are only 20 questions. And at the very least, you can have 0 questions right. So this distribution is discrete, and it's bounded in 2 directions. And if it's close to either of those values, it's going to be asymmetric, and then a normal approximation is just really not good. Yeah? Last one, we haven't used this during GRS, and I don't intend to use it during basic statistics for master students, but I just want to show you this is another very commonly used distribution from the exponential dispersion family, which is the gamma distribution. The one shown here is a special case called the exponential distribution, and well, it shows exponential decay. Yeah, so we can measure the time it takes until a light bulb burns out. The moment we turn on the light, it can burn out. So it cannot happen before we start measuring, because then we would take another lamp to measure. So again, it starts at 0, and yeah, it has a certain decrease, but the gamma distribution is very flexible. It can take on any kind of shape, as long as it is a strictly positive, continuous outcome. Yeah. So if you think, okay, my outcome cannot possibly be negative, and I know it's continuous, and I know it might have right skew or something, maybe try the gamma distribution. That's how using a GLM works. It's just which of these is more, makes more sense in the context of my research, and then you try that one and you do the model diagnostics to see if it is indeed an appropriate choice. Okay, so what is then the big subject that we're going to cover in this course, in the advanced statistics course, so not basic? That is the mixed model. And as I already said several times now, it allows you to model dependent data. So I already gave this example where you measure people at several time points. So yeah, if you measure the same people, then those measurements are correlated. If you start with a very high blood pressure, then you're going to end up slightly higher, probably. And if you start very low, then the next time point is probably also a bit lower than the rest. Yeah, so people have their own baseline measurement, and the correct way to deal with that is with a mixed model. Now, yeah, if you only had 2 time points, there is a very simple version of this that we did cover during GRS, and that's the paired t-test. Yeah, so you can think of the mixed model as the extension of a paired t-test to any kind of linear regression. Okay, second example. This is a nested design. In this case, we're not interested in the islands. We're interested in counts of birds on 2 types of terrain. So, yeah, excuse my drawing skills, but the green is forest, and the, well, what is it, yellowish is beach. And we want to know, is there a difference between the number of birds we observe on the beach and in the forest? But, you know, if one island is particularly rich in fruits and seeds, then of course there will be more birds. So the number of birds we're going to observe is nested in the island that we measure. And this kind of hierarchy, that's typically also something you can deal with in a mixed model. Yeah, because measurements taken on the same island, again, are correlated. Finally then, this is not something we're going, okay, I'm saying this, but I'm not the course coordinator of advanced statistics, but I think this is not something you're going to do because this is quite a bit more complex. But you can also use mixed models for spatial autocorrelation. So imagine that thing shown on the right, that is a plot of land, and you're a farmer. And you want to know which of 5 treatments is most effective on reducing herbivory, eating of your plants, basically. And you know, A, B, C, D, E. You have 5 treatments. Which one prevents herbivory best? Okay, so this is a Latin square. I've randomized them in 2 directions. So every row contains every treatment, every column contains every treatment, and it's random. So that's great. But still, let's say there's a river going over here. Then all the measurements that are near this river will probably have a different value than the measures that are further away from the river. So in spatial autocorrelations, measurements that are literally physically closer to each other will be more strongly correlated. The reason I say we're not going to do this, you can of course come to us if you need this for your internship, but when you do this, you have to specify a covariance structure. You have to tell the mixed model how are those measurements correlated. Are they only correlated to the ones next to each other, or is there a effect where slightly further correlates a little bit, slightly further, a little bit less, a little bit less, a little bit less. There are many ways to do this. You can make it incredibly complex or incredibly simple, and that's just, it's too much for the already very compact 4 weeks that you have. So we don't do that. I just wanted to show you these are the things you can do with a mixed model. Okay, yeah, so again, just think of it as the equivalent of the paired t-test for any kind of regression model. Now then, yeah, the last thing you're going to learn in advanced statistics that is still regression is the generalized linear mixed model, the GLMM. So now we can pick any distribution in the exponential dispersion family for the outcome, and we can have any combination of dependent and independent data. That is the big thing that you're going to learn in this course. Yeah? And if you can already give the answer to, okay, let's say I have measurements of this, and I've measured it like that, which model would be appropriate? If you can answer that, that is more than enough for this course. That is, yeah, that is about the level of difficulty. Okay, so just to reiterate, yeah, you can choose any suitable probability distribution from these, except gamma probably won't be used in the course very much, and you have any combination of dependent and independent data. Now, how you model it is something that, of course, we're going to teach you, otherwise it's a bit useless, but it is also a bit more difficult, so we will use it in the practicals, on the exam, we're not going to tell you write in R code how you would model this mixed model, because it's very easy to get it wrong, and there's multiple ways to argue for it. So the important part is that you understand this conceptually. Okay, and yeah, just to reiterate, you can use this with everything you already learned in the bachelor course. You can apply that to mixed effects models. So continuous, categorical, doesn't matter. Interaction, also possible. Transformation, you can do all of that. You can combine all of that. So that is the reason that we teach you, that we have such a heavy emphasis on linear regression, because if you pick the right things from what you learned, from transformation, from probability distributions, from dependent independent data, interaction, yes or no, you can make a very, very versatile model. You can do any number of things. Okay, so how then do you choose the right distribution? I'm going to go over this quick, but I just wanted to show you how you could argue for it. So when would I pick the normal distribution? When I have a central tendency, yeah, and when I have a continuous outcome, and if we're going to be very strict, then it should be a variable that can be both positive and negative. But again, all models are wrong as long as it's useful. You know, when we look at the height of human males in the Netherlands, I mean, how many standard deviations do you have to be away to cross 0? That's going to happen with such a low frequency according to the model that it doesn't matter that the model is wrong. It's a very nice approximation within the range of our data. Then the Poisson distribution is for independent counts. Count is anything that you count, so a number of this, number of that. Then binomial distribution is for several things, for binary data, so yes or no, or are you male or female as outcome in this case. Or ratios, so let's say I want to know whether a certain type of mask is more efficient than another type of mask, then I could have two groups of people wearing two types of mask, and I'm going to see how many of those people in the one group got infected and how many in the other group got infected. And that's a ratio. And you can do that with a binomial distribution. And the last one's tricky, it's very annoying, but it's very common in census data, so government government counts, which are often a hidden ratio, which means that it's of the form of, okay, this many people had infection, and then it says in small numbers per 100,000 inhabitants. But if you divide it by the number of inhabitants, it's no longer a count, it's a ratio. Yeah, so if it says the number of this per the number of that, that's a ratio, not a count. Then finally gamma, again, don't pay too much attention to this, we're not going to use it, but I just want to be complete. The gamma distribution is for non-negative continuous outcome, which usually has right skew. So one way you can model right skewed continuous data is with a gamma distribution. Then the last one, negative binomial. I'm going to turn my head towards Halt. Are you going to use this?
Yeah, well, in the case of overspreading.
Okay, yeah. Great, because that is what I've written down. The Poisson distribution assumes that every count is independent, and that assumption is actually very strict. Even when your study design is such that the counts should be independent, it's often still the case that there is some unmeasured influence, and then they're no longer independent counts. And usually what happens is that they're over-dispersed, and you can use a negative binomial model. Yeah, so if you didn't follow the third year course, I expect this to be a lot of information at once. That's fine. These are the things that we're going to cover slowly over the coming 4 weeks. If these things ring a bell, that should be enough for you to follow the advanced statistics course. Yeah, so again, you don't have to remember all of this. There will also in advanced statistics be a recap of GLMs of 2 days. And yeah, in basic statistics, this is the main subject, so we're going to spend a lot of time on it. Okay, now how do you account for dependent data? I just told you with a mixed model, and specifically by adding random effects to your model. But yeah, I'm not going to explain that now because we're going to discuss it at length, and it's a long answer. So if you want to know the answer now, it's it's over here. In basic statistics, we don't use mixed models because it just becomes too much, especially when you combine them with GLMs. It's tricky to work with. So in basic statistics, only GLMs. Okay, I just wanted to, give you full disclosure that in fact not everything is linear. So in gray, again, the GAM, generalized additive model, is not part of the course, so that's why I made it gray. But okay, that model, if we're going to be very strict, is still linear actually, but it is no longer a parametric model. So a generalized additive model is a large sum of the same model again and again and again. It tries to find different combinations to get to its conclusion, and it allows for many more distributions than the ones I just talked about. So it's very useful, but not part of the course. Then, so that one is non-parametric. It has no pre-specified form. That's what non-parametric means. Then the random forest, finally, is really a non-linear model. So it works by combining decision trees, and a decision tree is a non-linear model. Why do we cover random forest? Because they're really easy to work with, and they are very powerful. They often outperform really complex predictive models, even though it's super simple to learn how to use a random forest. Now, then neural networks. We think this is a very important subject to know a little bit about, because it has such a major impact on society at the moment, and also research. It is very difficult to use, because there's not because it's conceptually very difficult, because it's still a form of regression, just no longer linear and no longer parametric. But it's really versatile. You can do many different things. The thing is, though, you have to learn, the first thing you should really learn is how to know that your model is actually doing its job. That's something we do cover in the course. But then for neural networks, you have to make many choices. And combinations of choices quickly grow to a very large number. So yeah, effectively exploring what we can do with neural networks in a 4-week course, just where we already cover a lot of other things, is not going to be very nice. Small but also important reason is that most of your laptops are going to have a really hard time if we use neural networks that do anything remotely interesting. Yeah. I also made a little note here because I call these predictive models, but I just want to be clear, any model can be used for prediction, right? Just some models are better suited for inference, because under certain assumptions you can make really cool conclusions. And other models, like the random forest neural network, are essentially a black box. We don't really know how it came to its conclusion, but they do work really well for prediction. So that's why I call them predictive model. But yeah, the little side note there is that you can use any model for prediction. Okay. Finally, there are some things that are not even models, but we do learn them. So yeah, unsupervised learning, like PCA. Simulation, very useful. Most students, first time they hear about simulation, are like, okay, why would I want to make fake data? I'm not a teacher, so But it's actually also very useful in study design, because if you don't have data yet, how are you going to convince someone how many samples you need? If you have to go to an ethical committee and you have quite a complex design, so you can't just go to online power calculation or sample size calculation, then how can you meaningfully make an estimate of how many samples you need? And the answer is by making fake data, and making fake data with small variance, making fake data with large variance, and then seeing, okay, what is the effect on the required sample size? And then you can show the results of that to an ethical committee or to whoever you are going to convince that you need money or samples, or money for samples. So that is why simulation is really important for study design. Now, then there's multiple testing correction. Yeah, also important. It's essentially when you do more than one test, you need to do multiple testing correction. Then there's cross-validation, which is a way to assess whether your model is doing its job. Then there's bootstrapping, which is a way to estimate standard errors and confidence intervals in models where that is usually really tricky. Then the last subject we're going to cover, that's not really a model, in one of the last lectures by Harold, is gene set enrichment analysis. It's an example of a specialized technique, but it's very common in genomics, which is a kind of chi-squared test, but it's more like a whole method where you do different things. So it's not really a model. Yeah, so the reason I'm showing you this is to let you know that if you are just good at the regression part of this course, that covers most of it. That should, okay, I'm going to carefully look at all of it. That should be enough to pass the course. Maybe not with an A or a 10, but it should be enough, because the other things, you know, are just not something we talk about too much. Okay, Harold already told this, but it is very important that you make the right choice. So I am also going to give my own interpretation of when you should choose for basic or advanced. Basic statistics grew as a courtesy for students who are not from Leiden, because everybody has different backgrounds, and statistics in other educations is often taught by mathematicians, and mathematicians are, I mean, I studied math, so I'm also a mathematician, but most mathematicians, they feel like you should be able to do this by hand, and you should understand the mathematics behind it, and we would be spending most of the course, you know, deeply understanding something, you know, quite simple, like linear regression. And we don't really like that, because you are going to do an internship after this, and you are going to have to use this. And because of that, we want to cover a broad range of useful models and not delve too deep into why it actually works. If that does interest you, there is a master in statistics at Leiden University, but I think for most people, you chose this education because you like biology. And in that case, the really, really important thing to learn is just, what do I need? And, you know, it's nice if you can do it yourself, but the most important is that you know what you need, and also that you know that before you collect your data, so that you know how to design your study such that you can, you know, draw a meaningful conclusion from it. Okay, then finally in basic statistics, we also have to cover some useful techniques that are just very common, and a lot of the more difficult exploratory techniques are based on some version of PCA. So PCA is a very basic tool. It's just how to summarize very large data. So that's also something we talk about. Yeah, if you choose basic statistics, you have to pass advanced statistics next year. I'm just going to repeat this one more time. You know, you do get credits for basic statistics, and it is perfectly possible to follow advanced statistics next year and not have a study delay. That's the reason we have 2 courses. Yeah. So that's what you would choose basic statistics for. In advanced statistics, yeah, I mean, you have to do it if you want this degree. Also, the thing that we're going to do, the main thing we're going to do is just expand this regression framework to more and more and more problems, eventually to the GLMM. That's where we stop. There is more you can do, but yeah, we only have 4 weeks. We're going to talk a little bit about predictive modeling and how you can validate your model. And we're going to talk about some useful tools that are a bit too much for basic statistics. Okay, so regardless of which one you choose, I'm going to give you some tips. Some of them are really obvious, others maybe not, on how you can pass this course. The first one, sorry, it's obvious, but you have to avoid missing lectures or practicals. You know, there will be courses that are easier or courses that are more flexible. During your internship, if you can't come on a Monday, it's not as big a deal as during the advanced statistics course. It has this name for a reason. We have to cover somewhat interesting models for your internship. So again, we want each and every one of you to pass, but the learning curve is steep. You have to keep up. So if you can, try to be here. Second one, when we introduce a new technique, try and see how it relates to the previous subjects. And spoiler alert, most of it is linear regression. Okay, then, when we introduce a technique, the 2 things that you should focus on is, what is this method for, and how do I interpret the outputs? Because those are the things that you will need on the exam. You know, how it works is really useful to know, because all the assumptions are derived from how it works, and you know, when you can use it is derived from how it works, but that is secondary. For us, the most important thing is that you know what you need, and that you know how to interpret what it does. Yeah. So the rest is important to study at some point if you're going to continue a scientific career. But for the purpose of this course, it's secondary. The important stuff is how it works. Sorry, not how it works. The important stuff is what it's for and how you interpret the outputs. Yeah. So it also means you don't have to be some star programmer in R at the end of this course. Yeah. On the exam, you will get questions where we already did all the programming and you have to interpret it, and not you have to tell us how you would program something. That's not going to be part of the exam. Okay, finally, we try our best to post videos pertaining to subjects before we have a lecture on that subject. You don't have to watch those, but you know, of course, that will make it a lot easier for you to follow the lecture. And also, the second time you hear about it and there is an actual person standing in front of you, you can ask questions. So that might be useful. I'm going to check, speaking of questions, where they're already. Okay, yeah, there's a question. Is it required for the binomial distribution to be bound by both sides, or is it possible to be bound by one or no side? No, it is a ratio. So, and a ratio of things cannot be negative, and it cannot extend beyond the number of trials. You know, the binomial distribution is the number of successes out of n trials. And you cannot have more successes than trials, and you cannot have fewer successes than 0. So it is by definition bounded in 2 directions. But how far that bound is, that depends on your data. Okay, yeah, back to this. Yeah, oh yeah, ask questions. So this is especially important for people who are not from the Netherlands. Dutch culture is almost certainly different than your culture, but one important thing is that it is never wrong to ask a question. There are no stupid questions. There are no questions that are too easy, and you don't have to wait until you know the right time to ask a question. When you have a question, ask your question. It can be while I'm talking. Just please raise your hand. Can be before the lecture, can be after lecture, and in week 4 you can ask things about week 1. I don't care. I just want you guys to pass the course. So ask questions. Yeah. The other thing is where to ask questions. We really prefer that you use the Brightspace Q&A. I will quickly show it. Yeah, so on Brightspace, I'll just show it from here. If you go to Advanced Statistics, there's Course Tools. I hope this is visible. Let me make it a little bit bigger. Yeah. So if you are at the Advanced Statistics page, go to Course Tools, and then go to Discussions. Yeah, and I've already set up one for Advanced Statistics and one for Basic Statistics. But for this week, you know, we all do the same stuff, so it doesn't matter where you post it. You can just post in Advanced Statistics. I try to answer this whenever I can. Yeah, when I don't, it's either because I don't know the answer or because Harold has to answer it. Yeah, the other thing, there are of course questions that don't fall into this framework, but Harold already talked about it. If you have something personal or something you know you don't want to share with others, of course you can email us. If you don't have access to Brightspace, of course you can email us. But for anything else, yeah, it's just you know allows us to read each other's questions, so you can learn something from it. Also allows you to find questions by topic. So if you're like, well, I didn't really understand GLM very well, that's not really a question, right? A question is how would something specific work. And if other people already ask questions about that, that's really a nice way to learn actually. And that's also a way how I learn about new things within statistics, just by reading people's questions and yeah, expert answers. Then finally, yeah, it allows us to simply answer more questions, answer them in more depth. Because if we're not just addressing one person, but you know we're talking to the forum and everyone can view it, then that allows us to invest a lot more time in it, and it also prevents a lot of duplicate questions. So it's more efficient. Okay, finally, we are developing an online version of this course. Over here at ibl-inspire.github.io/advanced statistics. I will quickly show it. Yeah, so I already did this for the 3rd year course. Yeah, you know, we're trying to do a lot of things at once, so this is still a work in progress. Sorry, one moment. Yeah, we are making an online version where you can see every day's content. So I will try to, for example, over here, there's one, you know, the lecture study design. The practical is we're going to draw a map of coronavirus cases. And you can already see it. If you want to have a look beforehand, you can do it. If you want to look at it 1 year later, you can do that. That's the idea. And also, everyone who doesn't have access to Brightspace surely has access to internet, I hope. And they can go to this page. So that's the idea why we're doing this. For accurate information, always go to always have a look at the Brightspace announcements. That's the primary source for us to contact you. And you know, people who don't have access yet, we will try to email you the important announcements separately. But we try to include everything here so that it's just easier to find everything. Okay, sorry, you had a question?
You mentioned in the previous page that there are videos going to be uploaded. Is it in
Yeah, it's gonna be both. So, oh yeah, sorry, let me do this. Yeah. Okay, so I'll repeat the question just in case someone in another room didn't hear it. Whether the videos, so about this bullet point, whether they're going to be uploaded to YouTube or to Brightspace or to the sites I just showed. And the answer is all three. Of course, they're going to be uploaded to YouTube. That's the only way for me to share them. Then I post a link on the content of that day on Brightspace, and I try to copy it to the GitHub page. So everywhere is the answer. Yeah? Let me check if there were other questions by now. We are almost at the end. Yeah. Okay. This is the last slide. I'm going to look at the time. Harold, how much time do we have?
All the time in the world.
All the time? Oh, okay. Well, then let's do the exam.
Wait, let's see. They look quite fresh.
Yeah? Okay. Yeah. So, okay. So I have listed a number of research questions, and just like emails that we get from students, they're a little bit vague. So they are open to interpretation to some extent, but that doesn't matter. The question here is whether you can think of the right way to analyze this. Now, if the answer to all of these is yes, then we can go to the exam probably, but this is just to show you if this type of question is answerable for you after this course, then it was a success. Yeah? Some of these methods you have already had. So yeah, let's just give it a go. How about we have a short break, and then I will explain how to install on our studio, but first I will go over some of these so you have the break to see if there are any where you have an idea how you could analyze it. How long shall we do, Harold?
Maybe not that long. I mean, they all already have the whole day, so we have to be a little bit careful.
Okay, let's do a 10-minute break. We'll start at 3:40, and then I'll answer these questions, and I will also explain how to install the software. Okay, I'll now mute the stream and put it back online after the lecture, after the pause. Okay, the meeste mensen die most people who want to still follow the last part are here, I think. So let's continue with the last part of this lecture. Guys, shall we continue? Okay, I want to address two things. First, let's see if anyone can guess any one of these. And I want to address one of the questions that was asked. It's like, why can a model have more than one effect and still be linear, right? So I don't know if this is readable, but it says y hat, so the estimate value for y is equal to an intercept plus a slope times x plus a slope times x squared, right? So this is a simple polynomial model. Now the answer is, let's say we make, okay, I chose the wrong colors, but oh yeah, I do have blue. So, okay, it's always bad to use, okay, that doesn't. Well, it used to say X. Okay, well, let's just continue like this. Anyway, the intercept is wherever the outcome starts, right? When both explanatory variables are 0. So that's this point over here. Then we have the first effect, which is a slope, right? So a constant increase in a certain direction. Now, then we have the second explanatory effect, and just imagine a three-dimensional space. I can go linearly in one direction and still at the same time go linearly in another direction. This is what I tried to draw here, but I don't really know why I did that because I have a figure on it. Oh yeah, here we go. Yeah, so in this case, the systolic blood pressure, imagine you were age 0 and weight 0. You know, all models are wrong, so that doesn't actually make much sense, but imagine you were. Then this model says your systolic blood pressure would be here. And then for every increase in year by 1 year, the systolic blood pressure increases like this. And perpendicular to that, we have a linear increase with weight, right? So for every kilogram increase in body weight, we expect on average an increase in systolic blood pressure. Okay, is this model realistic? I don't know. But it is a linear model in two directions, right? So that's how something can be linear when you have more than one effect. When it's a polynomial model, yeah, okay, now we have the same effect squared. So it looks a bit different, but if we just call this x squared, we call it z, or we call it x2 or something, then you can see that it's really just the same case as this. It's just going to look different when you plot it, but the interpretation is the same. Were there any other questions from the previous part? No? Okay. Then let's go to the end. Yeah. So we have questions 1 to 9. Does anyone have an answer to any of these questions? You can also type in the chat. I'll have a look after the first ones appear. Yes, go ahead.
Maybe 6 because the x is either 1 or 0.
So what do you think it should be?
Normal. Oh wait, no.
Okay, so you can choose from the methods that I listed here. Okay. Let's see. Where was that? Yeah, anyway, like t-test, simple linear regression, multiple linear regression, etc. Oh, yeah, this. Yeah. Yeah, and when you say GLM, you should specify which distribution. Yeah, but other than that, that's the ones you can choose from. Yeah? So, anyone have an idea for any of these, how you could model that? Yes?
Okay, I don't know for that for one linear regression.
Okay, good guess, because most things are linear regressions. But, okay, so let's have, she says it's probably linear regression, always a nice answer, but for number 1 specifically, the crop yield in kilograms is compared for different concentrations of insect spray. Well, if I try to draw this study, it would look like this. Well, it will look like this. Yeah. Okay, so we have a concentration here. I don't know if this is visible, so let me quickly move this explanation to the center. Sorry. Yeah, we're just going to draw them all and then we'll see that the answers are that they make sense. Yeah, here we have the concentration, and here we have the crop yields. Yeah, so crop. I'll just write crops in kilograms. Yeah, and we expect that I would hope that the farmers are using these treatments to increase crop yields. So you would expect a positive increase with concentration, right? So maybe this, or maybe adding more and more and more is not really going to help anymore. So maybe a logarithmic effect. So this would be, okay, this is supposed to be straight. Y equals beta 0 plus beta 1 times x. Yeah, y hat. And this one would be y hat equals beta 0 plus beta 1 times log x. Yeah, but they're both simple linear regression because there's one explanatory effect. Yeah?
Why would you use crop yield and not crop loss?
Well, because I'm not a farmer. Yeah, no, I mean, of course you can model crop loss.
But isn't that more important for the thing you try to infer from it?
Yeah, I mean, it depends what you're trying to look at. If this is an, okay, I said insect spray, so you're right. Crop loss is more a direct way to model it. But crop yield, maybe insects are not the main thing limiting your crop yield. So that's, yeah, you could do both. Yeah. And another reason why you need both knowledge of biology and of statistics to set up a study efficiently. Yes?
Well, I'd say that crop loss might be difficult to model since you can't really weigh the amount of kilograms that have been eaten by the insects since they fly away.
Yeah, that's a really good question. I don't have the answer. Maybe she does. Yeah, so sorry for everyone listening. The question was why not crop loss instead of crop yield? And yeah, I don't know. Good addition. Yeah. Okay, anyone else for any of the other ones? This was number 1, by the way. In the meantime, I'm going to make some space. Number 3, ANOVA. Okay, so again, the crop yields. So we expect a continuous outcome with maybe central tendency or not for 3 different insect spray. Yeah, so number 3, we have 3 different means that we want to compare, or 3 different location parameters more generally, which you could do with ANOVA. And yeah, then after that you have to check whether ANOVA is appropriate, you know, whether normality holds, etc. But as a starting point, you could use ANOVA. So what would this study look like? So this is number 2. It would look like this. Again, we have the crops. Yeah, I'm just writing crops because it takes less time to write. And here we have treatment A, treatment B, and treatment C. And then, yeah, I would probably summarize this with a box plot or something. Yeah. And here we have 3 groups we're comparing, and if indeed it looks somewhat like this, then you can say, okay, this is a reasonable central tendency, you know, the mean is more or less in the middle, and then you could do this with ANOVA. So this is ANOVA, and the first one, if I can find black, is simple because there's only one explanatory effect, linear regression. Oops. Yeah, I'm not right. Okay, anyone has an idea for any of the other ones? Yeah.
Would 2 be a t-test?
Number 2, the height of trees of the same species is compared on 2 types of soil. So she says, could it be a t-test? I agree. If you compare the height of trees, and we're going to assume that, you know, all of the heights are somewhere above 0 meters. So it's not close to, if it's really, really close to 0, then the t-test is not going to work very well. But if they're all adult trees, then, you know, there's going to be some constant difference by the soil type. And then you could perfectly analyze this with a t-test. Yeah. Yeah, so it would look the same as this, except there's only 2 groups. Yeah.
5 of their t-test.
Okay, so number 5, tree density is compared before and after conservation policy changes. And she asked, could this be a pair t-test? And that's correct. So the more general way to do this would be with a mixed model, but since there are only 2 time points, before and after, with no other information specified, this could be perfectly done with just a simple paired t-test. Yeah. Yeah, and again it would look like this. Except the outcome would be tree density, whatever that means. Yes?
And maybe 9 in COVA.
Number 9, crop yield in kilograms, so continuous outcome, same one as the first two, is compared for different combinations of types of insect spray and concentration. And your answer was?
On COVA.
ANCOVA, yeah. So analysis of covariance is when you have an ANOVA and you have a continuous explanatory variable. So what are the continuous and categorical explanatory variables here?
The concentration
Is the,
compared to the kilograms.
so which is which?
You can continuous.
Yeah, so a continuous outcome would be the concentration, right? Because you can pick any value in between other values, so it's continuous. But type of treatment, well, it's either one or the other. So that's a categorical variable, and when you have that combination, you can use ANCOVA, which more or less looks like this. So here we have the crops again. I was very creative, as you can see. And here we have, let's just plot the concentration, yeah? But instead of having one line, we have multiple lines because, let's do linear for ease of interpretation now. Treatment A, treatment B, treatment C. Yeah? This is what ANCOVA looks like. We can immediately refresh everyone who did follow GRS their memory by saying, what would interaction look like? Does anyone have an idea? This is an ANCOVA. So, this is number 9. ANCOVA. Sorry? Yes, exactly. So, let's say that it looks like this. See, that's called interaction because the value of the slope for concentration depends on the other explanatory variable. Depends on which treatment. You know, for this it's this slope, for this it's that slope. That's called interaction. Yeah, if you don't know that yet, no problem. But if you did follow GRS, I hope that rings a bell. Okay, any other ones? If not, okay, yeah, go ahead.
Number 8, binomial.
Number 8, a binomial GLM. Okay, the number of deaths is compared between vaccinated and unvaccinated people. Okay, why is that your answer?
Oh, because it's ratio.
Yes, very good. So this one is, I phrased it in the annoying way that government census would do it. When you see number of deaths, if you did follow the GRS course, the first thing that should come to mind is it's a count, right? Number of death. But it isn't the number of deaths. It is the number of deaths per 100,000 inhabitants. And that's a ratio. And ratios can be modeled with a binomial GLM. Yeah? I'm not going to draw it because it takes a bit longer. But that's how you could analyze it. Okay, you can think on it a bit longer. In the meantime, I'm going to wrap up because the other ones, we can discuss it tomorrow or later in the course. We will repeat these. And what's always useful is if you know how to do one thing a certain way, then if you just switch the words with whatever is going on in your internship, then you can do the same thing for your internship. Yeah, so you can model a lot of different problems with linear regression in case it wasn't clear yet. Okay, so last thing is how to install R in RStudio. I'm not going to explain it here. It isn't very complicated. I just wanted to say, there's this video, if you haven't seen it yet, of how to install it. And there's a video on how to use the interface, what everything in the interface means, the second one. I really recommend that you also look at the second one. You don't have to look at the first one if you already reinstalled everything. But the second one, it shows you, for example, how to turn off inline output and how to set certain settings the same as on my computer and on Hall's computer, and everyone who uses this a lot. And then it will, it will prevent a lot of situations where we don't really know what's going on, especially, if we do part of it online. It's just easier if we all use the same settings. And if I say the console pane, that you should know, okay, that's the bottom left one. If I see the help pane, It just says how to look at the interface. So yeah. But yeah, I'm sure that you are all able to install R. Please do install it. Also, if you already have it, install it again, just new. Yeah, if you run into any trouble, Harold and I will be here for a little bit longer. If you want to do it at home, fine. Tomorrow morning we can also fix errors. If you want to fix errors, you can post on the Brightspace Q&A, or tomorrow morning you just come to us and show what's going wrong. Yeah? Okay, well, it's 10 to 4. There's a question. Yeah. Okay, let me repeat that because it's relevant for other people. So, is there a separate uses code for basic statistics? Harold, I think not, right?
No.
Wait, could you speak in the microphone? Yeah.
I also got several questions regarding if some, say for instance,
If you have no more questions, you can quietly leave.
if you do a course like business and biology, business. I have to check this, but it's possibly that you don't need to do advanced statistics. There are some other stuff also, things like the question just posed. That what was the question again?
There is a separate code for basic.
Oh yeah, separate code for basic statistics. I have to check that, but basically is you enroll for advanced statistics. All the information on both courses is there. How we organize that in uses, I will discuss that. We'll put that in the announcements. Yeah.
Okay, so the stream eindigen.
Yeah.
Okay, I'll end the stream now. Thank you all for attending. Ja, no new
Kijk, die andere
questions.
even sluiten.
Ja, is goed.
Toch even hier